In this last session of chapter number 2, now we are going to talk about the biopotential electrodes. In the earlier video, we had discussed about the various types of bioelectric signals that are being produced from the human body. Now when the biopotential signals that are being produced from different parts of the human body are very different in nature. For example, the potential that is to be measured from a cell is very different from that is to be measured for maybe ECG. So for every kind of measurement, we need to have a specialized or a specific structured electrode that can be used for making these biopotential measurements. So the electrodes that are being used to measure the bioelectric events of the human body, they are classified into three categories: microelectrodes, skin surface electrodes, and needle electrodes. The microelectrodes, they are used to measure the bioelectric potential near or within a single cell. So the tip of these electrodes is made up of such a scale that it can actually penetrate a single cell without damaging it. Then we have skin electrodes. These are popular, very popular, that we see around. These are used for ECG, EEG, etc. Okay, then we have needle electrodes. These electrodes are actually penetrated just below the surface of the skin, and they are placed for recording EEG signals from the local regions of the brain, or EMG signals, that is the signals coming from a specific group of muscles. Okay, so we'll just discuss them one by one. The first category that we are going to talk about is microelectrode. Now, microelectrode are the electrodes with the tip sufficiently small to penetrate a single cell in order to obtain the readings from within the cell. The tip of the electrode must be small enough to permit the penetration without damaging the cell. Okay, now this action usually becomes complicated due to the difficulty in accurately positioning the electrode with respect to the cell, right? So commercially available microelectrodes, they have two types of constructions. One is the metal electrode, and second is the micropipette structure. The metal electrodes, they are formed electrolytically. The etching is done, that is the tip of the fine tungsten or the stainless steel wire is etched electrolytically on to a desired size. Okay, and that particular edge is actually placed inside the cell, and the metal ion interface takes place where the metal contacts the electrolytes either inside or outside the cell. Now when we are talking of measuring the potential, and we have ionic potential at the cell level, so it is important that we have to design an electrode in such a way that it reaches the micro level, the cell level, and electrolysis should take place over there. That means a cell ion metal ion interface is important. We need an electrolyte, we need a metal ion. So electrolyte over here is actually the fluid that is present inside the cell or outside the cell, wherever we are making the measurement, and metal of course is the tip of the electrode that we are going to place. Okay, so the second type is micropipette. It is actually a glass pipette of suitable size with the tip drawn out of the desired size, and that tip is actually filled with compatible cell fluid. Okay, so in that case, the ions that are present in the fluid already, they come in contact with the suitable electrolyte that is filled in the glass pipette, and the electrolysis takes place, which gives rise to the potential that we can measure. The second category is the most popular one, that is the skin surface electrode that we see around. So the electrodes used to obtain the bioelectric potential from the surface of the body are found in many sizes and many forms. Okay, although any type of surface electrode can be used to sense ECG, EEG, or EMG potentials. However, the larger electrodes are usually associated with ECG, since localization of the measurement is not important in ECG, whereas the smaller electrodes are used in EEG and EMG measurements, because in EEG and EMG sometimes very specific position is required while making the recording. Okay. Now, these skin surface electrodes that were, the earlier models of these skin surface electrodes that were designed, they suffered from many limitations. Okay, they were all most importantly, they were all very sensitive to movement. Even the slightest movement, caused the changes in the electrode potential and the impedance. In many cases, the potential changes were so severe that they completely used to block the bioelectric potential that the electrode was actually trying to measure. Okay, so to overcome these issues, the design was modulated a little bit and floating skin surface electrodes were designed. The principle of this electrode is to practically eliminate the movement artifact by avoiding any direct contact of the metal with the skin. The only conductive path between metal and the skin is the conductive paste or the jelly. Okay, the performance of the electrode is not impaired as long as the conductive paste maintains the contact with both the skin and the metal. In the diagram, you can see that the metal disc is actually, inserted into an electrolyte gel. There is a gel placed over there, and that gel actually is coming in contact with the skin. So earlier, what used to happen is that if slightest movement was there, the skin and the metal electrode would get disconnected, and there would be a disruption in the measurement. Now what happens, since it is a jelly kind of structure, it is comparatively robust to movement. So as long as the connectivity is maintained, that is skin connected to electrolytic gel and gel connected to metal, as long as this path is maintained, the signal would not get disrupted. So floating type of electrode surface electrodes now started becoming quite popular because these they are not affected easily by movement. Okay, and in due course of time, for ECG we had spray electrodes, disposable electrodes, and many different types of designs have commercially come up these days. Then you can also have, for skin surface, if you need some localized measurements, then we can have certain specific design electrodes, like we can have an ear clip electrode. You can see in the picture that, one is the ground electrode, one is the reference electrode, and like a clip, you can actually put it on your earlobe. Similarly, from the scalp, if from the skin you want certain measurements, then we can have skin surface electrodes, which can be actually placed on the surface of the skin, near the to the brain. Okay, these are not generally used, but these are specifically designed for special measurements. Then we have needle electrodes. Now, to reduce the interface impedance and other movement limitations, small subdermal needles are used to penetrate the scalp for EEG measurements. Okay, and even EMG. These electric needle electrodes are not inserted into the brain, they are merely penetrated in the skin. Generally, they are simply inserted through a small section of the skin just beneath the surface and parallel to it. Okay, so very small needles are there, and they are, whether you want an EMG measurement or you want an EEG measurement, a slight insertion in the skin is made, and just beneath the surface of the skin, parallel to the skin surface, these needles are actually placed. Okay. Occasionally, in animal and human research, longer needles are actually inserted into the brain to obtain the localized measurements of potentials from a specific part of the brain. This process requires longer needles precisely located by means of a map of the brain. A special instrument called stereotaxic instrument is used to hold the animal's head and guide the placement of electrodes. These electrodes are often implanted to permit repeated measurements over an extended period of time. In some research applications, simultaneous measurements from various depths in the brain along with certain axes is required. Special electrodes are developed for such application, which consists of a bundle of thin wires, each terminating at a different depth. You can see in the figure that we have a cluster of wires, very very thin wires, and these are actually guided inside the brain, and the wires, they are of different lengths. So the tip of the wire is actually placed at different depths of the brain, and that can give you a sustained measurement of various localized regions of brain. So this is not normally used. It is basically more utilized when you are doing research, or you might want, very long-term measurements are required. Maybe some extreme cases of coma or something like that. Okay. The needle electrodes for EMG, they consist of fine insulated wires such that their bare tips are in contact with the nerve muscle or other tissues from which the measurement is to be made. The remaining portion of the wire is covered with some form of insulation to prevent shorting. The wire electrodes of copper and platinum are often used for EMG pickup from specific muscles. The wires are either surgically implanted or introduced by means of a hypodermic needle that is later withdrawn, leaving the wire electrode in its place. In this type of electrode, the metal electrolyte interface takes place between the insulated tip of the wire and the electrolytes present in the human body. The needle electrodes that create the interface beneath the surface of the skin seem to be less susceptible to movement limitations than the skin surface electrodes. By making a direct contact with the subdermal tissue or the intercellular fluids, these electrodes also have lower impedances as compared to the surface electrode. So sometimes for very special kind of measurements in EEG, we can have needle electrodes or we may have skin surface electrodes. Similarly, for EMG, you might be using skin surface electrode or you might be using the needle electrode depending upon, how from what depth we actually want to take the measurement, right? So these are basically three categories in which we can classify the various electrodes that we use for measuring the different biopotential signals. Right. Now, another important electrical parameter is skin impedance. Right. The human skin actually acts like a variable resistance due to the activity of sweat glands. Now, we all know that the skin, it contains sweat glands, and sweat is nothing but saline solution, which is a good conductor of electricity, right? So when the sweat glands, they are empty, the skin resistance is high and the conductivity is very low. But when a person sweats more, the sweat glands, they are filled with sweat, the conductivity increases and the resistance decreases because the sweat actually is a very good conductor of electricity. So skin resistance measurement method is connecting the electrode sensor to two adjacent fingers of the object to record the resistance signal, and then amplifying, collecting and recording the signal by some amplification circuit. In the figure you can see that you have connected two electrodes to the adjacent fingers, and with the help of a multimeter or any device, a recording device, you can actually measure the resistance signal between the two fingers, right? So this is actually called as galvanic skin response. The resistance response of the skin is actually called galvanic skin response. It refers to changes in the sweat gland activity that are reflective of the intensity of the emotional state. Now, it is very popularly used in methods like lie detectors, etc. It is believed that when a person is supposed say lying or is under mental stress, so in that case a person sweats more, the sweat glands, they are filled with saline solution, and the conductivity increases and the resistance decreases, so the amount of current that flows actually increases. Okay, so skin impedance is another very important factor that can be used to measure emotional states, that can be used for skin treatments, that is dermatological treatments, etc. Okay, and even sometimes in psychiatric treatments we use this particular parameter. So we have lots of potentials developing inside human body, and we have specific type of electrodes that are designed to measure these types of bioelectric impedances.